As Jay said, I've been with Zoetis for the last about 2 years, and I have had the opportunity to work on the PED project, and here specifically the conditional license, and I'm going to talk with you about some of the studies that we ran to support the conditional license for our inactivated PEDV vaccine. So we received our conditional license in September of 2014, and as you may know, this vaccine is an inactivated whole viral vaccine. So it contains all of the relevant proteins for inducing an immune response, and this is also an adjuvanted vaccine. And as part of that conditional license, we are working forward with the efficacy and potency testings that are necessary to support the full license of this product as well. I wanted to take just a minute to talk a little bit about the differences between a conditional license and a full license. As we know, PED appeared in the U.S. in April of 2013, and since then has dramatically and rapidly spread across the U.S. Within just the last few months, we have had another state that's become PED positive. So this brings us to 35 states in the U.S. that have experienced PED. Of course, PED is in Canada, Latin America, and other countries globally. The impact is variable because of different strains and different virulence patterns of those strains as well. In June of 2014, the USDA issued a federal order which mandated reporting as well as implementation of a reasonable herd management plan of the swine enteric corona diseases. And really because of all of these reasons, the USDA, and specifically the CVB, or the Center for Veterinary Biologics, approved a path for conditional license for a PEDV vaccine. And what the conditional license really allows is for companies to bring products to producers quickly, especially in these emergency situations. For a full license for a vaccine, a demonstration of efficacy in the target animal is necessary. So this involves, in most cases, a challenge study. And for a conditional license, a reasonable expectation of efficacy is the requirement. Also, for a full license, a completely validated potency assay is needed, and for a conditional license, those potency assays need to be in development. The safety testing is the same from a conditional license to a full license, and this involves doing safety evaluations in animals spanning the age of those that would be using the vaccine, as well as spanning geographic locations, and this is to account for differences in genetics, management style, and things of that nature. We also conducted a field safety and field efficacy test, which is beyond these requirements, as well as a sow booster study looking at responses in sows at a time that would be consistent with our recommendation for a booster vaccination. So moving right into some of these studies, the first I'm going to talk about is our expectation of efficacy. And this is a serologic study, so we did not do any challenging as part of this study. The objective was to demonstrate a reasonable expectation of efficacy, and this was from the CVB guideline to have at least fourfold difference in PEDV-specific antibodies in piglets from vaccinated sows compared to piglets from non-vaccinated sows. You can see our study design. We have 4 different treatment groups, the first treatment group being the control animals, and then treatment 2 through 4 being 3 different vaccine formulations. All of these animals received 2 mils of their respective treatment, IM, at 5 and 2 weeks pre-farrowing. We collected serum from sows prior to each vaccination as well as 7 days post the second vaccination. And we also collected serum from piglets when they were 3 days of age. These sera were sent to SDSU for FFN, or neutralizing antibody testing. And the piglet serum, these are 5 piglets, 5 randomly selected piglets per litter. And as we do with all of our studies, we also evaluate safety of our vaccines. So this is looking at injection site observations, adverse events, general health, and whenever there are sows and farrowing involved, we also look at farrowing information and reproductive outcomes. These are the results of our expectation of efficacy test. In this figure you can see this is our FFN antibody testing results, and this is in piglet serum again. So these piglets are 3 days of age at the time of this blood collection. This orange bar is corresponding to piglets from control animals, so control litters. And these other 3 bars are showing the results of the FFN antibody testing from piglets of the vaccinate treatment groups. And you can see that these FFN antibody titers in these groups were significantly greater than that of our control group, and these were actually greater than 7-fold compared to those in our control group. We did not observe any adverse systemic reactions or adverse local reactions or negative impacts to reproductive outcomes either. So we did induce FFN antibodies that were induced in sows. These were transferred to piglets, and we were able to detect these antibodies in piglet serum. So here we have established a reasonable expectation of efficacy. We wanted to take this one step further, however. And what we did with this next study was actually maintain these sows from this last study at our research farm. We maintained them on site for 4 months because we wanted to booster them at a time that was consistent with our recommendation for boostering prior to a subsequent farrowing. So these animals were not bred, so all of the data that we have are actually from those sows themselves. This figure is showing the FFN antibody titers in sow serum following that booster vaccination. This is geometric mean FFN titer, here again in our serum of control sows, and then in the serum of our vaccinate sows. And you can see that at 7 days post-vaccination as well as 14 days post the booster vaccination, that we do have a strong and rapid anamnestic response. And in fact, these responses were at least 4 to 6-fold greater than what we had in our initial 2 doses of the vaccination. So conclusions from this study is that revaccination 4 months post the initial vaccination schedule did induce anamnestic antibody responses, neutralizing responses in vaccinated sows. So we have a reasonable expectation of efficacy, but what we really wanted to do was to bring this vaccine to the field to see how it would perform in a commercial setting with commercial animals. So of course in most sow herds you would have harmonized immunity on all of those animals. But in order for us to evaluate the efficacy and safety of this vaccine, we did have 2 groups of animals. So we have an adjuvant placebo group of animals as well as a group of vaccinate animals. And we had 120 sows and their litters in each of these groups. Similarly, all of these animals were vaccinated at 5 and 2 weeks pre-farrowing. And there's 2 important things to note about this study. The first is that this was conducted in a commercial production facility, and in late February, this site experienced a confirmed PED outbreak. We initiated this study in March, and between the time of that PED confirmation and the initiation of our study, these animals were given feedback at 2 separate times. The other thing to note about this study is that it is a generalized randomized block design with rolling enrollment. And so what that means is 8 subsequent weeks we enrolled animals. And we enrolled 30 animals at each of these weeks for 8 weeks. So if you think about our vaccination interval of 5 weeks plus the farrowing to weaning interval of 3 weeks, the first set of data that we're going to have looking at pre-weaning mortality is 8 weeks post-study initiation. And then also because of the rolling enrollment, which happened for 8 weeks, the actual last data that we have as part of this study is 16 weeks post-initiation of this study. We did confirm the presence of PED on the farm by PCR testing of pooled fecal swabs from piglets. So this was 3 to 5 piglets per litter, and the litters that we tested were randomly selected from these different blocks. And these fecal swabs were collected at the time of processing, and because of the conditions on this farm and their management, this was at approximately 10 days of age. We did evaluate safety by general health observations in sows and piglets, as well as injection site observations. Our main efficacy outcome was pre-weaning mortality due to PED. So moving to our results, I don't have the antibody result testing on here, but even though these animals were clearly not naive to PED, they did have neutralizing antibodies in their serum. After we vaccinated them, we did see a clear booster response. This top figure is looking at the back-transformed mean percent litter pre-weaning mortality due to PED. Sorry, that's kind of a mouthful. So I do have these data separated by parent class, so gilts, P1s to P3s, P4s, and then all. So this is all of the data summarized together. The orange bars are corresponding to our control litters, whereas these blue bars correspond to vaccinate litters. And again, this is accounting for that entire 16-week duration of the study as well. So in our control group, we have 6.3% litter pre-wean mortality due to PED, whereas in our vaccinate group, we have significantly less mortality due to PED. So this is 0.6%. And this is also a 90% relative reduction in pre-wean mortality due to PED. Also important to note is that vaccinated sows had 72% fewer litters with 100% pre-wean mortality compared to controls. And if we look at overall litters weaned, so that's this bottom table, in our control animals, only 78% of those litters actually weaned a pig. Whereas in our vaccinate litters, we had almost 94% of those litters that weaned a pig. Similarly to what we've seen in our other studies, under these conditions, this vaccine is safe. So vaccine administration, the vaccine was administered by farm staff because we wanted this to be as close to commercial conditions as possible. We didn't have any abnormal systemic reactions, didn't have any negative outcomes on reproductive performance. Looking at the economic analysis, so vaccinated sows had an additional 1.8 pigs survive per sow furrowed. And if you assume for a second that the average cost per viable pig born is about $31, 31 times 1.8 pigs gives you a direct savings of almost $56 per litter. If you assume an average profit per pig sold to market of $73 and account for a 25% birth to finish mortality, this may result in an additional profit of almost $100 per litter. So this is 1.8 times 73, accounting for this 25% loss. So in conclusion, the PEDV vaccine is safe for use in commercial animals. The vaccine is effective at helping to reduce the pre-wean mortality, even in the face of an acute outbreak. And we have shown an economical benefit of almost $100 per litter under the conditions of this study. So in conclusion, we are working to meet the requirements for the full license. So our potency and efficacy studies are ongoing. The safety testing, however, is complete, as the requirements for the conditional license and full license are the same. And of course, we are continuing to evaluate the impact of vaccination on disease control and looking to see how our vaccine can be used as a tool to help reduce the pre-wean mortality and PEDV disease. And I do think that based on these data that I'm sharing with you today, data that I don't have time to share with you, and our customer experience, that this vaccine really is a valuable tool to help reduce pre-wean mortality and PEDV disease.